So the three different types of RNA are mRNA, tRNA, and rRNA, and you're going to actually see them all in action in the next few minutes. mRNA stands for messenger RNA, and it's basically a blueprint that goes from the DNA to outside the nucleus to the rough ER where it goes to a ribosome. That brings me to the next one. An rRNA is just basically a ribosome. And it's a temporary little protein / RNA that comes together and is able to make those proteins. It needs the instructions from the blueprint. And then also the tRNA, or the ribosome, or tRNA, is called transfer RNA. And what these guys do is when the code says to bring a certain amino acid, it brings the amino acid and drops it off. So let's look at all these different things and what they do. mRNA is basically just a single-stranded message. It's got those guanine and adenine and cytosine, and the only one that's new now is uracil. And it's going to be made from the DNA, sent outside the nucleus to the ribosome, and these letters will tell us what nucleotides to read. And remember how we said we read amino acids in codes of 3, and those codes of 3 are going to be called a codon. A tRNA is just going to be something that, don't worry about the shape, but basically what it does is when there's something down here that says, hey, go ahead and get the specific amino acid, it'll go get that amino acid and bring it to the ribosome, and we'll see how to do that in a second. And a ribosome basically is where the mRNA will come in, and the ribosomes will come and start to build that amino acid chain. Remember that primary structure that we talked about in our first, one of our first videos? It's starting to build that. And just recalling, transcription occurs in the nucleus, and it's going from DNA to RNA. Translation occurs outside the cytoplasm, and it occurs from the mRNA making instructions to go to protein. It happens in the ribosome, and it's helped out by the two other types of RNA. That's the ribosomal RNA and transfer RNA. So let's go over transcription. So transcription occurs when we go from DNA to RNA. It occurs in the nucleus. It has some specific transcriptional factors that help the process get started and a promoter. The big difference here is when we were making DNA to another strand of DNA, we had this thing called DNA polymerase. But now that we're making RNA, we're just using this thing called RNA polymerase. And it's going to go ahead and make DNA into RNA. We also have that helicase that unwinded the DNA. So let's go over that process. So here is your DNA. We have that RNA polymerase, and basically what's happening is we're unwinding the DNA just a little bit, and that polymerase is looking at one side and saying, oh look, here's an A, here's a T, we're going to put an A, here's a we're going to put a U. And you can see that now it's starting to build a single-stranded piece of RNA. Our first step is going to be initiation. So where the RNA polymerase will go to the promoter, and it'll actually start to open it up and do that coding that we just saw. So we're just going to start to put the nucleotides in, except keep in mind we're using this new nucleotide, uracil, and we're not using thymine. The thymine will just be in the DNA, and the copy, that's the pink one, is the mRNA. That one's only going to have uracil. We're going to start to do this thing called elongation, where the polymerase is going to go down the DNA strand and start to copy this mRNA. And then finally what's going to happen is we're going to get to a stop sequence, and the RNA polymerase will stop. The mRNA is now the code that is going to code for a certain protein. and that will leave the nucleus. There are some editing that occurs before it does leave. We check for errors, we remove certain parts of the mRNA, so we do this thing called splicing, where we take out some of the garbage and keep all the good stuff together. Good DNA is called exons, and bad DNA is called introns, and introns actually get cut out. I'll show you how that happens in a second. And then before it leaves, just so the RNA doesn't degrade very quickly, we put a protective cap. It's called a 5' cap. It's like a helmet. And then a 3' tail, and that's going to protect the genetic material at the end. So here's your DNA, and it's mixed with these exons and introns. Introns are about 90% of your genetic information that you carry. The exons are the really good stuff. And we intermix the introns everywhere just to protect our genetic information. And we want to make sure that if there is any damage, by spreading out the really good stuff, more likely it's going to happen in the introns where we don't really care. So the first thing that we do is we remove the introns, and you can see we're cutting out, I guess it's the light pink color, that's going to be the introns, and the dark color the exons. We're going to cut out the introns, we're going to put everything together, and then right before we leave, we put a little cap and a tail, and those are designed to basically protect the mRNA and make sure that it's stable. So in our next video, we'll do translation.